Welcome to this edition of Feedstuffs FoodLink. I'm Sarah Muirhead. Nitrogen fertilizer has long been a part of profitable corn production. It also has been a large expense for producers. So what we're going to do today is take you to the University of Illinois, where there's a scientist who's looking at how we can perhaps rebuild the corn plant so that it will fix its own nitrogen, much like soybeans do. Let's go find out what it's all about.
We're working on a new field of engineering called synthetic biology, and it's sort of analogous to synthetic chemistry. Turn of the last century, when scientists went from just understanding atoms and molecules to actually putting them together to create new molecules, we got textiles and plastics. We're at the same juncture with synthetic biology now, where we can think about putting together genes and gene regulatory systems to create biological functions that haven't existed. So we can take inspiration from plants like soybean. Soybean knows how to engage in a molecular trade with its root ecosystem, with the microbiota, with the microbes in its soil, and trade with them and have those soil microbes fix nitrogen as a service in return. So we ask, why can't we teach corn how to do this?
By combining the science of microbial ecology with the tools and techniques of synthetic biology, novel solutions such as nitrogen fixation may be possible.
So this is enabled by two things: our understanding of the soil microbiology as regards to soybean, as well as our ability to modify some of these microbes and the plant material.
For farmers, this new technological approach could eventually translate into a cost savings.
The nitrogen fertilizer is an expensive thing. It's energy expensive. It takes a lot of energy to convert nitrogen to ammonia, and energy is going to become more expensive, and certainly anything we do to help reduce those costs of inputs is going to help the American farmer. It's going to help American agribusiness. Secondly, this technology need not only be applied to corn. We can think about other things as well. So for developing nations where they do not have access to expensive fertilizer, with these technologies we can help developing nations as well.
Environmental benefits may also result from the application of synthetic biology on the farm.
When you're not using all that gasoline, all those petroleum products to create nitrogen fertilizer, instead outsourcing it to the corn plant, you're saving all that energy. And so that has a direct impact on our climate, on the amount of pollution that's caused by petroleum products, and hopefully converting that into a biological process is a more benign thing for the environment at large.
The issue of synthetic biology and its potential is being brought front and center by the scientific community at this time in order to create an understanding and support of what's ahead. In fact, it could be a decade or longer before commercial application of any products from this system are achieved.
We are actually at the very early stages of this. Just to give you an analogy, we're like inventing the transistor. We cannot foresee, we're beginning to foresee what the internet of this technology is going to look like. I think the main focus of our lab right now is in understanding how, when we do modify organisms, when we genetically alter them, we want to understand how it affects the organism itself, because that is key to understanding how our perturbations affect the native system, whether it is the native ecosystem or the corn plant and what have you. And so by understanding the molecular burden of a genetic circuit on the cell, we hope to define rules that allow us to then place limits on how much engineering we can do with these systems, how much can the plant tolerate, what can we get away with.
If indeed a way can be found to convince corn to fix its own nitrogen, more corn-on-corn acres could also be a possibility.
And the whole idea behind corn-soybean rotation was that soybean fixes part of the nitrogen, and then hopefully corn can utilize that. Now the other reason is that it also puts a break in the pathogenic cycles between corn and soybean. So that's the other advantage of doing crop rotation. And maybe for that reason we might still continue to have crop rotation. I don't know, that this particular approach might help us solve the pathogenicity problem. But certainly making corn more efficient at fixing its own nitrogen and even utilizing it. And then the question is, why stop at corn? You know, if it is a generic enough technology, then we can do the same thing with wheat, we can do the same thing with the rice, and we can do the same thing with maybe potatoes, and it generally opens up the field to addressing a lot of different issues. And then why stop at nitrogen? Once we solve that, we want to go on to potassium and phosphorus.
It may also be possible at some point to use synthetic biology to change the nutritive value of a grain.
Really too early to say, but if you look at legumes in general that can fix their own nitrogen, they are higher in protein content by the virtue of them being able to utilize nitrogen more efficiently and fix their own nitrogen. It ends up as amino acids and as protein in the plant material. So yes, it probably will change the composition, but I think we might also be able to develop technologies to dial down or dial up some of the nutrient levels along the way. Synthetic biology is a very powerful concept. It talks about not just, we think it's what the future of biotechnology is going to look like. We think this is how genetic engineering should look like rather than what it does look like right now. We're relying on mathematical models, on well-developed theories. We're relying on genetic parts, the development, characterization, standardization of genetic parts, the traceability of these genetic parts, so that we can reuse some of these components from species to species.
Synthetic biology is a fast-growing research area with a wide range of potential applications.
We have biological components, genes, gene regulatory systems, and then we hook them up together just like Lego blocks, and we create whatever comes to the imagination.